What is this Parkinson disease?
Parkinson's disease is a progressive neurodegenerative disorder which manifests mainly as movement disorder. So often it is predominated by these motor symptoms. However, with research, there's been immense number of non-motor symptoms such as hyposmia, constipation, sleep disturbances. It has been reported that they occur at an early stage of the disease. So by the time the clinicians get to diagnose, there is minimum of 60% death of dopaminergic neurons in the substantia nigra. Dr. Ravi Yadav, he is specialized in REM sleep disorder, and we also have a sleep clinic over here run by Professor Bindu Kutti. So places like NIMHANS, these can be evaluated, experts' opinion can be taken.
Yeah.
So even if the dopaminergic neurons are alive at an early onset, they can show a functional disturbance, a lessening of these dopamine release. I would say again over there, calcium imaging, electrophysiology through MIA are important parameters to state that indeed that those cells are showing a deficit for the dopaminergic neuronal function.
We are predisposed to genetic disease like this.
So genetic testing has been now been referred by clinician neurologists, even in India. Though the occurrence is much lesser than the idiopathic, but there are minimum of 14 genes which are identified now as at-risk genes, be it your LUX-GIANT, alpha-synuclein, DJ1, Parkinson, PINK1, emphasizing it's a very complex disease and it's very heterogeneous. The heterogeneity is one of the reasons why many clinical trials are failing. For LUX-GIANT worldwide, especially funded by Michael J. Fox Foundation, there was good amount of studies being performed for LUX-GIANT. iPSCs as model?
Yes.
No, it's still a little rare. Yeah.
Parkinson's is a complex and multifaceted neurodegenerative disease that affects millions of people worldwide, causing motor symptoms like tremors, rigidity, and bradykinesia, and as well as non-motor symptoms like cognitive impairment, depression, and anxiety. Welcome to the Somnath Show. Today, we are joined by Dr. Indrani Datta, an additional professor in the Department of Biophysics, NIMHANS. Her research work focuses on Parkinson's disease, spanning from its underlying etiology to the development of potential therapeutics. Central to her work are the proteins alpha-synuclein and LUX-2. While not limited to dopaminergic neurons, her study extends to understanding the role of niche cells and how the neuronal microenvironment is altered under pathological conditions, with an aim to modulate it for therapeutic benefits. In this podcast, we will try to understand what are the symptoms of Parkinson's disease, the manifestations, research developments, and probable therapeutic options available which can change the lives of millions of people who are affected by this condition. Before we start with today's episode, it's my request that please like, share, and subscribe. Your one subscription can immensely motivate me and my team. So, on that note, let's get started with today's episode. We'll start today with the Parkinson disease. So Parkinson disease is one of those mental problems which many people are facing. And why I'm saying mental, because I just to set the context of discussion, because most of my audience will be within the age group of 25 to 35 and majorly will be also like common people. So we'll start with layman's language and then gradually we'll deep dive into your like work and to get an understanding of it. So ma'am, what is this Parkinson disease?
So, telling it in a very simple definition, Parkinson's disease is a progressive neurodegenerative disorder which manifests mainly as movement disorder. Now, it shows classical movement disorders such as bradykinesia, um, tremor at rest, rigidity, then postural instability. So these are the symptoms when uh, a patient or an individual understands that they have to visit a clinic or to a neurologist, and that's when the diagnosis starts out. So often it is predominated by these motor symptoms. However, with research, and be it clinical research or be it basic research, there's been immense number of non-motor symptoms which have been identified now, recognized also by clinicians, such as hyposmia, constipation, sleep disturbances, and several of them. One of the reasons why these non-motor symptoms, why I mentioned over here, have been in limelight is because it has been reported that they occur at an early stage of the disease, before the onset of the motor symptoms. So now, why is it important to know? So by the time the clinicians get to diagnose, already the patient's motor symptoms initiates, and research says that there is minimum of 60% death of dopaminergic neurons in the substantia nigra pars compacta. So this is a region in the midbrain which, again, in a layman's language, I can say it helps us to control the voluntary movement. Our voluntary movements are controlled by this part of the brain. Now, if the clinicians can diagnose it at a much earlier stage, and there are definitive non-motor symptoms wherein patients and individuals can report or start visiting the clinicians, then they would have a bigger window to intervene, be it through drugs, pharmaceutical drugs, or through interventions through many other supportive palliative care, or might slow down the disease progression, especially. So yes, presently I would say it has both motor symptoms and non-motor symptoms.
Okay. Ma'am, in India, what is the predominant of this? Roughly around.
Yes, it's a very good question and a big question of concern. So we don't have a big epidemiology data point from India with respect to stating this much percentage has been known to be diagnosed by PD, but I'm sure the NDS group from India is working on it to come out with this data, which is extremely important for even taking decisions in government policies. However, what we can see, or what we have understood from the evidences from clinics, be it hospital like NIMHANS, which is a premier institute of specifically for neurology. So there is a good number who have been diagnosed. And another thing in India, people have to also know and understand that, yes, I need to go to visit a clinician neurologist.
Right.
So I think this awareness that where they have to visit with respect to this movement disorder they are facing can also, it needs to be considered. So might be we are missing out also on those individuals because here the patient goes to the doctors. So we might be missing out the number there too.
Right.
And it also gets delayed for their treatment strategies to be initiated. But the number is quite alarming. And in general, we all know that age is a big risk factor.
Yeah.
Along with exposure to pesticides and pollution. These are big risk factor because 90% of Parkinson's disease is idiopathic. That is of unknown cause. So for these particular cases where we know that in our country the longevity is increasing, hence the occurrence of PD will be also increasing with it. And yes, we don't have a specific answer, but definitely it is a good amount of population who is suffering from it. And I don't think in near future it's going to be stagnant or going low till we don't take care of exposure to the risk factors, the environmental risk factors, to be more specific.
As you said, environmental risk factors is our lifestyle, the corporate culture these days, the stress, pollution, all this cumulatively have effect, because we see the rise in number of cases year on year now.
Right.
And so how much these factors contribute towards it?
Right. So one can be the awareness of individuals, of us in India, just reporting to the clinic. So that can also increase the number. But second thing is, definitively stress is one of the major factor for any immune-related disorders, any immune-related degenerative disorders to be particular. And Parkinson's disease, we know that the fulcrum of the disease progression is definitely showing that it is the immunological disorder which increases the progression. So that would definitely play a role. However, among the environmental factors, I can cite that evidence-based research has definitively shown that pesticides, exposure to pesticides by farmers, that is one of definitive cause for onset of Parkinson's disease eventually in life. So that's definitely a concern, and probably more education and bigger amount of epidemiological studies on these from India would help to also establish that farmers should be given to wear PPE before whenever they are handling pesticides and all. So it is not only awareness, but such epidemiology studies would also help in making government policy decisions.
Right. Yeah, well said. Well said. Because as you rightly said, earlier people were not aware. So the reporting was also might be not there that much. Yeah, that's one good thing is awareness is there. Coming to awareness again, if to a normal person, if I have to say, okay, these are the do's and don'ts, or these are the symptoms maybe for your identification at a very early stage at home, without any supervision of a doctor or going to a clinic, can there be any like 2 or 3 symptoms where we can just think this might be leading to Parkinson's?
Okay, so here I would say that I'm actually not certified for it because I'm a basic researcher, not a clinician. However, from the research papers which we refer and the evidence being present in literature, we can say that definitely hyposmia, sleep disturbances. So definitely if these two are present, and also along with it constipation, because it is both a brain to body, so a brain first body disease and also a body first brain disease. So the onset of the disease can be from the gut to brain or from the nose, that is through nose to brain to gut, to the body. So if these symptoms are being present along with fatigue, pain and cognitive decline or mood disturbances, a visit to a sleep clinic where a neurologist will be also present, and also to clinician neurologists would definitely help to define that whether it is particularly due to Parkinson's disease or just any other ailment, not with respect to PD. So, but it's not a decisive diagnosis that it would be, but definitely if there are especially sleep disturbances, it is advisable that you refer to a neurologist.
No, I think then many people should be going, because
Yes.
these days getting a peaceful sleep is becoming rare. As I talk to my colleagues and all, I know most of the people have sleep disorders. Knowingly, unknowingly, we are not aware of it.
Correct. So here, Dr. Ravi Yadav, he is specialized in REM sleep disorder
Wow.
with respect to especially neurological disorders, be it PD or MSA and others. And we also have a sleep clinic over here run by Professor Bindu Kutti. So yes, places like NIMHANS, there are centers where these can be evaluated and the expert's opinion can be taken.
Yeah, that's well talked about, because I think most people should take advantage, especially if you are in and around Bangalore.
Yes.
And NIMHANS is there as a wonderful place. And so, ma'am, again, like symptoms, early symptoms, but what if somebody comes to know at a later stage? Will that have some impact in recovery, or how is it like that?
True. It's very unfortunate for this particular disease. It's very complex. It's immensely complex, and that's one of the reasons why we do not yet have a cure for the disease. Impact, in the sense, till date, I would say even at the present date, most of the time it is diagnosed when the motor onset initiates, and they visit the clinicians. So, if the therapy or the drug interventions are initiated early on, yes, probably the severity or a progression can be little lesser. But as I'm saying, it cannot be generalized, because there are multiple factors and there is heterogeneity. There is immense amount of complexity. It is not that an intervention initiated earlier can bring about a lesser severity for all cases. However, the palliative care needed or the supportive care needed, help needed, those can help in the lifestyle of the person, the lifestyle leading of the person. It can be helping in managing the disorder a little better, be it with respect to cognition, be it respect to sleep, be it respect to your constipation, that's your gut problems, and be it respect to intervention through physiotherapy. So such things can be, as and when prescribed by the neurologist, such things would definitely help the individual to manage it a bit better. Yeah, but I won't make a statement that an early intervention always would help in reducing the severity or the progression.
Okay.
For some, there are evidences wherein dopamine, by Dr. Susan Fox, her group in California, wherein the dopamine intervention at an earlier stage has been shown to be correlating with lesser severity of the motor symptoms. So that's in US, the study is present. So with respect to that evidence-based study, I can state, yes, an early intervention would probably help in lessening the motor symptoms severity, but not halting the disease.
Right.
Yes.
So yeah, it's a nice point, ma'am, because we always have this perception, had I known this at the initial stage, this could have been solved, or this could have been treated properly, so that I would have not landed onto the entire manifestation of the disease, right? So this is a common thing which goes in mind of people. As far as I think Parkinson is there, and you cannot say it is like always true. Yeah. So MRI is the last resort to finally diagnose it, or
Yeah. So again, over here, I'm not the main person who can comment on this, but because I'm a basic researcher, but what I've learned from the clinician neurologist, so I was fortunate, and I am fortunate to associate with them as collaborators over here at NIMHANS. We're a very good team of clinician neurologists and scientists here. And I had to interact with them when I was going ahead for deriving the Parkinson's disease patient iPSCs.
Mm-hmm.
So therein the clinical diagnosis was extremely important with respect to be it your onset age, be it the severity, be it the UPDRS score for therein to actually define the severity. Along with that, we had Pal, Sir Dr. Professor Pal and Professor Ravi Yadav. They ensured that the fluorodopa PET imaging is done. So there can be movement disorder being initiated due to head injury or due to any other reason, but fluorodopa PET data is a definitive to confirm that indeed it is due to Parkinson's disease.
Okay.
And that's where I would limit to say that yes, the confirmation often is done over here by clinicians by using fluorodopa PET. So we have a very good radiology and imaging facility at NIMHANS department and facility. So yes, that's often used to many a times confirm the disease.
Right. Ma'am, for your like coming to a little bit on your research aspect, because that is where I would like to spend a little bit more time maybe. So when you induce, what type of model you use to induce Parkinson and study? So what do you use for it?
Yeah, it's very important for a basic researcher.
Yeah.
So I would say that we select the model based on a research question. So with respect to that, we start with even gain of function model if we are going ahead with genetic at-risk mutations. So the protein of interest wherein we are focused now are alpha-synuclein and LUG-2. So we use the gain of function model, be it in cell lines which have dopaminergic neuronal properties, or if you're working on astrocytes, then on astroglial cells. That's to understand that cause and consequence of a genetic problem or at-risk genes. However, with respect to the human relevance, it's extremely important to have the human Parkinson's disease patient-derived cells as a model. As I said, I was fortunate to work over here with the clinicians in collaboration. So we have generated the PD patient iPSCs carrying mutations such as LUG-2 or Parkin and PINK1, and also from sporadic Parkinson's disease patients. So that's definitely, I would say, a very big model for us to venture into the mechanism of the pathogenesis, and as simple as whether the PD phenotype is getting replicated in the Petri dish or not. So when we are stating that we have generated, we also can now state that it's not just a generation, but we have also seen the PD pathology being replicated in the dopaminergic neurons, which we differentiated from those iPSCs. And similarly, the astrocytes differentiated from those patient iPSCs. So that's so these are the two in vitro models we definitely work on for any research question. So we try to keep two experimental models to answer a research question. And with respect to in vivo, we have the chronic model, the MPTP-induced rat chronic model. Why chronic model is important? Because again, we work on the therapeutic part too. So we needed a chronic model to understand the translation effect of be it a stem cell derived drug delivery vehicle or stem cell derived cells itself. How does it function there? And just a brief why MPTP and RAC model we used, because usually if you provide MPTP into or administer MPTP in rat through intraperitoneal or through blood, it won't get PD symptoms because the monoamine oxidase P enzyme is quite high in their blood. So it cleaves it into MPP plus, and MPP plus cannot cross the blood-brain barrier. However, it's been proven well from a group in Brazil and been used quite a bit across the world. When you administer it through intranasal route, it brings about a chronic Parkinson's disease symptom.
Okay.
So initially the non-motor symptoms appear, and then the motor symptoms starts out. So we utilize that model too. So these are the three, this is the only in vivo model we have at present. Yes, we would always love to go ahead for other knock-in models for the at-risk genes we are doing, but as of now, we don't have the facility here.
Right. Once you have developed your model for that, then what are do you have a screening criteria for that? On what parameters you select that, okay, they have been PD case or not?
Right. So for each, so I'll start with the in vivo model. So initially we started with a standardization, and we had reported it also as literally a standardization data we have published. So we had done a dose dependent study, and from there we understood that one dose of MPTP is good enough to bring about initiate non-motor and motor symptoms. It takes time. The motor symptoms initiates by week 2 post MPTP, and from day 3 post MPTP, the non-motor symptoms initiates. But as I'm saying to you, it is not just by observation. So we do experiments with respect to hyposmia. Then we do the electrophysiology for the nerve conduction velocity as, again, a non-motor symptom. And then we find that, so this is a pre-event than the motor symptom. So the motor coordination by rotor rod, then locomotor by OFT, that's the open field test, and actimeter. So most of these are, they have IR sensors so that it's not relied on a manual counting. So those now instruments are available even in India and for our institutes to do these measurements, to understand that each time when we bring induce MPTP induced PD model, whether the rat of that group has got the PD symptoms or not. So it's a bunch of non-motor tests. And in the non-motor, again, we keep one which is very functional and electrophysiology based. And then in motor symptoms, again, the motor coordination and the locomotor through actimeter are very definitive parameters for us to state whether the onset has happened or not.
Right.
And again, coming to in vitro also, it is very important. It's not only in vivo to understand. So in vitro too, we utilize again the dopaminergic neurons. Their main function is they should be secreting, or rather the vesicular release of dopamine should happen once they're stimulated. So even if the dopaminergic neurons are alive at an early onset, they can show a functional disturbance, a lessening of these dopamine release. So that can be due to many parameters. I'm not getting into that depth. So those we definitely measure by the vesicular dopamine release, stimulation by light cell calcium imaging when we are stimulating through physiological stimuli too, and also by multi-electrode array. So I would say again over there, calcium imaging, electrophysiology through MIA are important parameters to state that indeed that those cells are showing a deficit for the dopaminergic neuronal function. And pathologically, we also look into the alpha-synuclein expression, whether that accumulation we are able to see more in the PD model, or the modified form, the phosphorylated alpha-synuclein expression of that is higher or not in those particular cell types, be it in vivo through IHC and here in vitro through immunocytochemistry. So that's, I would say, marker-based expression also confirms that, yeah, the protein of interest, which are being classical neuropathological markers, are also getting to appear over here.
Right. From all this point, can we say that PD is also genetically we are predisposed to genetic disease like this? We can say that.
Yeah.
Or is it too early to come to that conclusion?
No, there are many genes now being considered. So genetic testing has been now quite being referred by clinician neurologists, even in India. So as simple as classify them as genetic due to genetic reason or due to sporadic, that is idiopathic. And there are though the occurrence is much lesser than the idiopathic, but there are minimum of 14 genes which are identified now as at-risk genes, be it your LUX-2, alpha-synuclein, DJ1, Parkin1, PINK1, even GBA1. So those are so carriers of such genes, like might be the onset hasn't happened yet, but for most of these genes, the onset would occur.
So if I do my like lineage wise, like my father, mother, and their ancestors, if I do that screening, maybe I can come to a conclusion that today I'm not showing the symptoms, but it's quite likely with age this might come to me.
Yes, so it needs more research. You said it very well, because how can we say definitively, right? For Parkinson's, that's why I'm coming back again and emphasizing it's a very complex disease, and it's very heterogeneous. The heterogeneity is one of the reasons why many clinical trials are failing. Okay, it shows very good result in till the animal model, but it fails. So I'll give you one example, especially for LUX-2 in worldwide, especially funded by Michael J. Fox Foundation. There was good amount of studies being performed for LUX-2. Now, LUX-2 is one of the mutation which is most common genetic mutation worldwide. So it was being now what we understand is, or rather for the past 5-6 years, it's very clear that the variants, there are numerous variants present for LUX-2, that protein itself. So the one which was reported long back, like G2019S, it's predominant in the Caucasian population and the Ashkenazi Jews.
Okay.
And that has been worked elaborately and extensively. And kinase inhibitors, there are many studies on it which works very good for such patients. However, G2019S mutation, or rather the variant, is nil. It's negligible in India or in the Eastern Hemisphere. What we get to find from reports, again, individual reports, not in epidemiology studies, that the variants which have been reported mostly are in the GT-PACE domain, not in that kinase domain, which is the G2019S. And from India, we found it's still rare, which is I1371V, and which was through bioinformatics studies being denoted as non-pathogenic. So we often forget that it's a predictive tool, and if the clinically the person is showing classical PD symptoms, so there's something amiss over there which needs basic science research to established through evidence that indeed that particular variant is a benign or it is pathogenic.
Okay.
So from our study, so we did both a causation study, so cause and consequence study through the gain of function, and also from the patient derived iPSCs, be it the dopaminergic neurons, be it the astrocytes, and we showed that intrinsically it is a pathogenic mutation. So what I understand now in MDSI, that's the Movement Disorder Society in India, considers this LUG-2 I1371V as a pathogenic variant.
Okay.
Okay, so but for that we needed to do the basic science research, and we are delving more into it to understand how is it different, and that's why these variant carrying patients, they respond differently to kinase inhibitors. Not only to kinase inhibitors, there have been clinical reports, wherein it's been found that the deep brain stimulation does not bring about a positive effect for this I1371V variant. Rather, it impairs further.
Mhm.
On the other hand, it improves for the G2019S variant carriers. Similarly,
Yeah.
another very clinical observation is there, wherein I1371V, the onset age is earlier, and the Levi body has been found in the brain of this carrier. And Levi body reports with respect to the carriers of G2019S is rare. It's not being reported much.
Yeah. Okay.
So you can understand from the clinical data and the postmortem data that screams out loud that there are many differences between
True.
just a point mutation being different.
Correct.
And so we believe that basic science research is extremely important to eventually state that not only find out new drug targets, and also to establish why, as simple as this, the difference is present
Right.
between
Yeah.
a variant, a point mutation in the kinase domain, and a point mutation in the GT-PACE domain. And probably if we do further with the patient cells to validate our results, which we found in our causation studies, it would help to get, we can use the iPSC platform to have predictive population
Yeah.
for drug screening. And that would ensure better, clinical trial data.
Yeah. Agreed.
Yes, it
Absolutely.
would be quite, might be more positive results.
Would
will come
possible,
up.
and I think, ma'am, in India, where you have cohort to
So
cohort, the
big.
gene pools are very different.
Very different. And here only I wanted to mention it's not, so I1371V we still consider it to be a rare variant. The thing is that what we understand from the clinic data is that we cannot assign that one particular variant is higher being found in India. So rather the heterogeneity is more, because LUG-2 is a very big variant.
Yeah.
Okay, so there are many places where the mutation can happen. And when I'm stating here, the carriers have been reporting PD symptoms and they're responding to dopamine. So it's not that, we cannot just take it to be a benign, because the person carrier is showing classical symptoms of PD.
Correct.
So these variants, there are numerous of them, and it's a very typical, I would say, this particular occurrence of the heterogeneity, even within LUG-2, is quite an occurrence in India rather than in literature if you search in the West. So it makes it complex,
Yeah.
furthermore complex. Yes.
Yeah. Where variability is huge. The gene
So again,
diversity.
coming back to it, iPSC patient-based height
Yeah.
drug screening
Yeah. Correct.
is needed before a patient has been considered in a clinical trial for any LUX-GIANT
Agreed, ma'am.
medicine.
But as of now, how many, like roughly what population or what percentage of Indian populations has been screened? No data onto that, right?
We don't have a data. There's a study which is in preprint now with NIMHANS been also a group in that.
Okay.
So once it gets published, we can understand.
That would be good.
Yeah.
Yeah.
But definitely more bigger large scale epidemiology studies are also needed to track down people who are not yet even having PD, but who are the carriers of such genes. That would also help to understand the occurrence of it, because in India it is not always that it gets reported in the clinic.
Ma'am, we are talking about therapy and gene-based therapy, right? Like stem cells used for as a therapeutic for Parkinson disease. It's like I see pluripotent stem cells is what you have worked with. So iPSCs is what I saw in your this. So can you just briefly explain what this is all about?
Sure, that's also one of our not only a favorite model system, but so I would say that we started iPSCs. We work on cellular biophysics, to be more clear, because we don't do banking as of now for iPSCs. Our mission is to understand the mechanistic pathways which are bringing about this heterogeneity, or as simple as pathogenesis in different cell types for a patient, a mutation, particularly what we see in India. So with that, it's a big topic to talk about, but we started with the LARC2. Now, therein we are using the iPSCs as a model. My other lineage through which I come from, because I worked with mesenchymal stromal cells, and over there, we know that mesenchymal stromal cells are used even in clinical trials, and it's humongous potential for, because one thing is they are non-tumorigenic and they can bring about immunomodulation. However, it needs to be defined more how to go ahead with it with respect to any disease type. It's not if randomly and rushed and given and started, that's where no answer, it will come to a failure again. So understanding the disease, understanding the patient's pathogenesis, and then going ahead and targeting specifically what you want to target is extremely important. So with respect to mesenchymal stromal cells, we definitely are working with one of the products, that's the exosomes. So we use the biophysical method of isolating exosomes from human dental pulp stem cells. And one of the reasons is to use them as a drug delivery vehicle.
Okay.
So we have performed the in vivo studies and few of the in vitro studies, which it's under communication, it's under revision. And we have also filed a patent before that. So now I would say that we would be, we haven't yet started therapeutics or from iPSC derived neuronal cells or astrocytes. Yes. But we have initiated our work with exosomes, be it exosomes on its own as a therapeutic product, or them as a drug delivery vehicle for Parkinson's disease. That's where we have initiated. Yeah, but we are working. I won't be mentioning it here, but we are working on some other cell type too from iPSCs, wherein we think that it has, not think, or we have data now, we are making it stronger to understand that whether they have effect or how they bring about improvement in PD.
Yes. And then right now in India, roughly how many labs are working on this? On like how many labs are working? Roughly, I'm just to get an idea.
In Parkinson's disease, there are multiple labs which are working.
And mostly are working on the stem cell aspect of.
No.
Okay.
No. With respect to stem cell, with respect to using stem cells as a model,
Yeah.
iPSCs as model,
Yes.
no, it's still a little rare. Yeah.
Okay.
Yeah, it's still a little rare. Yes. As per the last PubMed search, I would say, yeah, I would go by evidence. It's still not there. Yeah, we got a head start, as I say, that because I was being fortunate to have a setup like NIMHANS. So it really helps to have clinicians on board, and where the collaboration works well. And yeah, we got a head start due to that.
So, ma'am, in this like Parkinson disease research group, there might be a lot of consortium or societies, India as well as internationally, right?
Yes.
So, what are they like? If you can name few of
Yeah.
them.
So one of the society, International Society, that's a Movement Disorder Society, is the largest, I guess. So I'm a member of that, definitively. So every year there is an MDS conference, which gives a very good platform for interacting, networking with clinician scientists and also basic scientists.
Okay.
So along with that, we also have a LUX-GIANT consortium, which I'm associated. That's a consortium among 3 countries, Luxembourg, Germany, and India. So presently, the work is, that's from that consortium work is where the preprint is with respect to the genetic mutations being, found in India in reference to PD.
Mm-hmm.
So I would say the work with respect to the clinical team is definitely at pace and it's going ahead.
Mm-hmm.
It hasn't yet percolated to the basic science, so it's going in stages. Once we know exactly that which is the prevalent ones and wherein the clinical questions are arising, we will be going ahead with the basic science projects from there. But it's a good consortium in the sense we get to learn a lot
Yeah.
and interact and understand the differences or the similarities, what we experience from a German cohort or Luxembourg and India, and how Indian PD cases are there heterogeneous or they're a little unique, and if so, in which parameters. So and the meeting happens every year, once at least in a year. Very face-to-face meeting happens once every year. So that gives a good bonding and understanding.
Also exchange of information.
Yeah.
And yeah, that's
Yes.
nice.
So these are the 2 consortium or society I would definitely mention. And there's another meeting which happens quite often. 2 meetings happens actually. One is Gordon Research Conference on Parkinson's disease, which I started attending from the lab. I think 2 years back it had happened. I'll be missing this year's because I'd attended the alpha-synuclein meeting at Cambridge. So I was fortunate to present on stage our work. So that's another, you would say, a society or consortium kind, wherein alpha-synuclein people working on scientists working, be it clinician scientists and basic scientists, we all gathered to network and learn from each other, again, exchange ideas, network, and understand that which ones are working, which is not. So it's quite a learning experience, definitely.
Yeah.
So these are the 4 meetings or societies which I definitely keep a track, or I try to attend them because it helps for our research question and our work.
So, thank you for agreeing to come here and then share out your your information, knowledge, and your work experience with us.
Yeah.
So with that note, thank you, ma'am.
Thanks a lot for this whole episode. Thank you.
Thank you.